we go. All right, so I'm going to talk about three different types of protein interaction strategies. I'm going to talk a little bit about affinity purification, proximity labeling, and chemical cross-linking. And so, especially given that this is, where we are at the end of the day, unless you want me to talk for an hour and a half, I really hope this is, turns into a discussion more than a lecture, because I have a feeling that that many of you have interest in doing pull downs and probably more expertise in doing some of these experiments than I do. And so, starting out with an affinity pull down or a co-immunoprecipitation, this is something that has it's been around for a long time. Before mass spectrometry really was involved, you pull down protein of interest, and then you run a gel and you do a Western blot of your known interacting partners to show that your protein is interacting in a complex with your targeted protein. And so wasn't a problem with if it pulled down some non-specific things, because you had a fairly specific antibody to pull down, a sensitive antibody anyway, and then you were probing with another antibody that was fairly specific, and so you wouldn't want to look at a stain gel from an affinity pull down, but you look at it by Western on the three proteins that are interacting with it, and it looks pretty good. These experiments don't work particularly well by mass spectrometry, and we do our fair share of them. A lot of people do them. The problem is that the specificity is just not there, and so you just end up getting a lot of proteins, and it's difficult to sort out what is a true interactor and what is just coming along for the ride. And so luckily in the around 2000, I think it was in 99, a group came up with a method that it's been become known as TAP-TAG or Tandem Affinity Purification, where you take multiple affinity portions in a fusion protein and a cleavage site in between. And so what was first used were two protein A domains, TEV protease and a calmodulin binding region, and you can do a first capture with IgG, and then you can hit it with a TEV protease to release it. You can take what's released and bind it to calmodulin, and then you can elute it off the calmodulin. I would show the diagram from the original paper, but those who are like me that remember 1999, we're just happy that you could get a color figure in a paper in 1999, and so the graphics aren't very good. This is a more recent paper, but just kind of shows the process a little better. But I do show the gel, and you can see how good the resolution here is. Apologize for the graininess, but this does show, effectively what this looks like when you do the pull down. If you remove one of either the calmodulin binding step or the cleavage of how complex these are, but if you actually use all three, if you use the IgG beads, if you cleave the protease, and then if you bind again to calmodulin and elute it, you can actually run a gel and see individual bands that show the complex. And so this idea of doing tandem affinity really is necessary for looking at interactions with mass spectrometry. And so we have a researcher at UAMS, Justin Lung, who does a lot of pull downs, and his favorite combination of tag is a flag tag combined with essentially an avidin linker. And so using both of those in combination, they get really nice pull downs. And so it doesn't have to be these two, but having two different pull downs allow you to bind and rinse, and then rinse again. And so that really is necessary to elute away and remove a lot of the false positive binding that you get. Proximity labeling is something that's kind of it's fairly new to us, and so described I guess in 2012 first, and it's a fusion protein. That's I think that says fusion. This picture may be in front of it, or maybe it's a fusio protein. No, it's a fusion protein that makes biotin, and essentially your protein of interest, when activated, will start making biotin, and it will biotinylate things in a fairly short region, so you can control this, the timing of this. Then you can do a streptavidin pull down or avidin pull down and get the biotinylated proteins, so you can at least look at all the usual suspects, all the guilty partners that are close by. Because it's going to label everything, this kind of gets a long list, and so there's lots of follow-up that needs to be done in terms of validation. But it is an interesting way to get to some labeling. And then finally, I'll talk a little bit about chemical cross-linking. And so chemical cross-linking has been around for a number of years. The reagents get better, the mass spectrometers have gotten better, and the software is getting better. And so it's really probably just about to I think hit its prime. And so one thing that this is nice, you can look at single proteins, where a certain protein may fold back together, and you can look at interactions, within a protein, within a complex, and a lot of these reagents are cell permeable, and so you can actually look at interactions, within an organelle or a cell or a tissue, and so typically the chemical cross-linker will have two functional groups, and then based on how far apart these are, it will allow labeling if two things are within that proximity. The majority of these use succinimide esters binding to lysine, just because that's chemistry that's pretty easy to do, and lysine allows pretty good coverage of a lot of interactions. And then there are a couple of different ways we can look at this using mass spectrometry. Early on, all of these were what we now call non-cleavable cross-linkers. So the chemical moiety, linked together two proteins, and then we would digest this, and in the mass spectrometer we would see that cross-linked peptide, and then when we fragmented, we got fragments from both peptides, and this is a problem because of the increased search space. It actually doesn't make the problem twice as bad. It makes the problem a squared as bad, and so it causes a lot more ambiguity in identifications, and it really makes it logarithmically more complex. There are cleavable cross-linkers, and there are a variety. There are photocleavable, acid cleavable, a lot of different chemistry. But basically what it allows you to do is to, a mass spectrometry cleavable will allow you to have two peptides that are linked. And in the mass spectrometer, you can fragment them and get the individual peptides, and then do another round of fragmentation within the mass spectrometer and sequence both of those peptides. And so it's this approach really kind of driven by the sensitivity of being able to do an MS3 experiment in a mass spectrometer that has made this a little more feasible in recent years. And so the one that we have done at least a little bit with is DSSO, and it's succinimyl sulfoxide. and so it is an NHS ester, so it's going to bind to primary amines. It has a length of about 10 angstroms, so it's pretty small. And so what the mass spectrometer does is it isolates a peptide and fragments it, and we can fragment this with just a little bit of energy, less energy than we would typically use to fragment, and the linker breaks, and so it gives you one peptide with part of the linker, that same peptide with a little bit more of the linker, and then the other peptide again with the full linker and with part of the linker. And so this mass difference is 32 Daltons, and so it allows us to kind of program the mass spectrometer to look for these. And so it's not as complex. Oftentimes when you're looking at these, you know, the vast majority of peptides in your sample are not cross-linked. And so the ability for the mass spectrometer to find this needle in the haystack is aided by the fact that there's a known mass difference between these. And then we can sequence those individual peptides and then use some tools to make the identification. And there are a couple of different fragmentation mechanisms that we can use. Still, there are difficulties here. One in that oftentimes if your concentration is not right, what you end up with are a lot of dead ends. So you'll see, you know, one peptide or protein that has the cross linker, but then there's not a peptide on the other end, or you get self interactions. And so you're only binding, you know, one protein to itself, which is not nearly as interesting as finding a binding partner. And so, much to Dennis's chagrin, I didn't talk for an hour and a half, but I really thought that this would probably be a point, just introducing that enough that maybe we can spawn some conversations on protein interactions, because that really seems to kind of hit home with a lot of researchers.